This is Chijioke Oramadike. I'm here to discuss how protein synthesis relies on genetic information. Protein synthesis. This amazing artwork shows a process that takes place in the cells of all human things, the production of proteins. It actually consists of two processes, which is the transcription and the translation. Transcription. During transcription, DNA is used as a template to make a molecule of messenger RNA, that's a mRNA. This is the first part of the central dogma of molecular biology, the DNA to the RNA. It is the transfer of genetic instructions in DNA to mRNA. During transcription, a strand of mRNA is made up to complement a strand of DNA. Steps of transcription. There are three steps that take place in transcription, which is the initiation, the elongation, and the termination. Initiation is the beginning of transcription. Elongation is the addition of nucleotides to the mRNA strand, while termination is the ending of transcription. That is, the mRNA strand is complete. Processing mRNA. When an RNA transcript is first made in a eukaryotic cell, it is considered a pre-mRNA and must be processed into a messenger RNA, which is the mRNA. In processing the mRNA, it may include splicing, editing, and polyannulation. Translation. This is the second part of the central dogma of molecular biology, from the RNA to protein. It is the process in which the genetic code in mRNA is read to make a protein. Translation is illustrated in the diagram below. After mRNA leaves the nucleus, it moves to the ribosomes, which consists of the RNA and proteins. Also, the molecule of mRNA leaves the nucleus and goes to a ribosome in the cytoplasm, where translation occurs. During translation, the genetic code in mRNA is read and used to make a protein. In conclusion, after a polypeptide chain is synthesized, it may undergo additional processes, or it may also bind with other different types of molecules, such as lipids or carbohydrates. Also, in summary, we've understood that protein synthesis is the process in which cells make proteins, and it occurs in two stages, which is the transcription and the translation. The transcription happens to be the transfer of genetic instructions in DNA to mRNA in the nucleus, while translation occurs at the ribosomes, which consists of the RNA and proteins. After a polypeptide chain is synthesized, it may undergo additional processing to form the finished protein. These are my sources, and thank you for listening.